National Institutes of Health researchers have isolated a set of promising, tiny antibodies, or nanobodies, against SARS-CoV-2 that were produced by a llama named Cormac. Preliminary results published in scientific reports suggest that at least one of these nanobodies, called NIH-CoV-112, could prevent infections and detect virus particles by grabbing hold of SARS-CoV-2 spike proteins. In addition, the nanobody appeared to work equally well in either liquid or aerosol form, suggesting it could remain effective after inhalation. SARS-CoV-2 is the virus that causes COVID-19. The study was led by a pair of neuroscientists, Thomas J. T. J. Esparza, B.S., and David L. Brody, M.D., Ph.D., who work in a brain imaging lab at the NIH's National Institute of Neurological Disorders and Stroke, NINDS. For years TJ and I had been testing out how to use nanobodies to improve brain imaging. When the pandemic broke, we thought this was a once-in-a-lifetime, all-hands-on-deck situation and joined the fight, said Dr. Brody, who is also a professor at Uniformed Service. On average, these proteins are about a tenth the weight of most human antibodies. These chips play a critical role in the immune system's defenses by recognizing proteins on viruses, bacteria, and other invaders, also known as antigens. For instance, a few years ago scientists showed that humanized nanobodies may be more effective at treating an autoimmune form of thrombotic thrombocytopenic purpura, a rare blood disorder, than current therapies. Since the pandemic broke, several researchers have produced llama nanobodies against the SARS-CoV-2 spike protein that may be effective at preventing infections. In the current study, the researchers used a slightly different strategy than others to find nanobodies that may work especially well. The SARS-CoV-2 spike protein acts like a key. It does this by opening the door to infections when it binds to a protein called the angiotensin-converting enzyme 2, ACE2, receptor, found on the surface of some cells, said Mr. Esparza, the lead author of the study. Test tube studies showed that this nanobody bound to the ACE2 receptor 2 to 10 times stronger than nanobodies produced by other labs. Other experiments suggested that the NIH nanobody stuck directly to the ACE2 receptor binding portion of the spike protein. Then the team showed that the NIH-CoV-112 nanobody could be effective at preventing coronavirus infections. To mimic the SARS-CoV-2 virus, the researchers genetically mutated a harmless pseudovirus so that it could use the spike protein. The researchers saw that relatively low levels of the NIH-CoV-112 nanobodies prevented the pseudovirus from infecting these cells in petri dishes. Importantly, the researchers showed that the nanobody was equally effective in preventing the infections in petri dishes when it was sprayed through the kind of nebulizer, or inhaler, often used to help treat patients with asthma. The team has applied for a patent on the NIH-CoV-112 nanobody. Although we have a lot more work ahead of us, these results represent a promising first step, said Mr. Esparza. Collaborators are also working to find out whether they could be used for inexpensive and accurate testing. Please support our channel to grow by pressing subscribe button as well as the bell icon for daily science updates. Thank you.